from the break at this point. See if I can get set up correctly. It's always a little bit of a gamble. If I can get my chat and my Q&A up at the same time, we'll see if I can multitask it. All right. So welcome back from the short break. If you left us, if you stayed with us this whole time, we had a little bit of discussion during the middle, tried to get to some of your open questions from the first half. I'll try to also do like a little bit of a review from the first half, a little bit more like kind of higher level, like we gave you a ton of information. The point of that is not that you should know every piece of that inside and out from, like from the heart, you know all of that memorized. It's more to be a reference for you so that as you go either into proteomics or more in depth into proteomics, these aren't new things that you have to Google. You'll be like, oh yeah, I vaguely remember someone mentioning that and it was a quantitative technique. That's kind of the idea. So in this section, I'm going to go a little bit more into data independent acquisition in the lecture portion. And this is to help set you up for tomorrow and Wednesday when Brendan McLean is going to be walking you through doing more in-depth Skyline for data independent acquisition. And then in the hands-on section, I dropped the tutorial download in the chat. The hands-on section, we're going to do a little bit more of a deep dive into looking at data and curating data in Skyline. So I will try my best to keep up with all of the various chat and Q&A, but Sue, Brendan, anyone, feel free to interrupt me if there's a lot of questions in the chat. So as I mentioned, we're going to talk today, this section, a little bit more about data independent acquisition. I'm going to give you a lot of information, but there's really kind of 4 key things that I hope that you take away from this lecture. The first is to just know some of the key differences between data independent acquisition, some of these other acquisition methods that we mentioned this morning. Second, I want to give you a little bit of exposure. There's a lot of different methods out there, but I just want to expose you to some of the various kind of flavors of DIA that you might come across. Then we're going to talk, we're going to spend all this time talking about how great DIA is, and I use it for everything. I'm going to tell you why there's caveats for DIA, and you might not want to use DIA for everything, either because of the way DIA is inherently set up or the way the data analysis is done. And then hopefully by the end of this, you just have the ability to kind of evaluate when there might be benefits for using data independent acquisition and when there might be limitations that make using, your classic TMT or your PRM or your SRM targeted analyses more appropriate. So this morning, to kind of summarize a lot of the techniques that Sue discussed, when we're choosing how to operate the mass spectrometer for proteomics, we're choosing 3 kind of things, and we're going to compromise something. The first is how scalable the throughput is. So that's how many samples you have to run. If you have to run 6 samples, or do you have to run 600 samples? That's going to affect the way you want to run your mass spectrometer. These slides will be made available right after this. I kind of, as we went this morning, I beefed up some sections that it seemed people had more questions about, and I took out other sections to make room for that. So it was a very dynamic process, but I'll get the slides up as soon as we're done with this. So scalable throughput, first thing we have to think about, how many samples are we running? Second thing we have to think about, how comprehensive do we need to measure the proteome? So this is how many peptides, how many proteins do we have to measure? Some research questions, you really only care about one protein or a handful of proteins. Other research questions, you want to know every single protein that's in the sample. And then finally, how sensitive of quantification do you need for your experimental purposes? Sometimes you might not really care. You just kind of want a general idea. Is this peptide more abundant or less abundant? Some questions are very clinical, and you have to be able to say with high certainty that there is, 5 picograms per deciliter of blood of this biomarker. So these 3 things are going to dictate which way you choose to run your mass spectrometer. You'll also be limited by what hardware you have, of course. If you have lots of samples and you need very sensitive quantification, but you don't need to measure a lot of proteins, you're going to choose something like targeted mass spectrometry. So this is things like that SRM, selected reaction monitoring, PRM, parallel reaction monitoring, or MRM, multiple reaction monitoring. So SRM and MRM, those are performed on a triple quadrupole, versus a PRM is performed on an ion trap, on an Orbitrap, that kind of instrument. So this is, you have a very limited number of peptides or proteins you're interested in. You want to measure lots of samples, or you want to measure very high sensitivity quantification. On the other hand, if you don't really care that much about the quantification, or you care more about getting kind of a survey, a list of all the peptides and proteins that are present or absent in your sample, you might do what's called discovery mass spectrometry or shotgun mass spectrometry. This gives you that list of how many proteins and what they are in your sample. You can do a lot of samples this way, and you'll get that protein list, but the quantification is not going to be particularly good. This is kind of those DDA-based methods. So finally, Sue mentioned these isotopic labeling approaches. So these kind of combine the ability to get that protein list with better quantification than if you just did a regular DDA experiment. So she mentioned things like TMT and iTrack. These are kits that you purchase, you prep your samples, you label your samples, you mix them together. Typically, you then fractionate. So even though you mixed them together, now you have the exact same number of samples that you started with. You measure them by DDA and analyze them with your software of choice. Lots of different software options for DDA. The downside of doing this isotopic labeling is typically you're kind of capped at like 16 samples. So if you need to analyze 600 samples, for example, this might not be your best choice. So we're going to talk today about data independent acquisition. A lot of people in the mass spec proteomics field, including myself, feel like DIA is a good compromise between all of these. You can measure hundreds of samples, you'll get the full list of all the proteins that are in that sample, and you get very sensitive quantification compared to some of these other methods. So we're going to visualize DIA using these kind of ion maps. So in these ion maps, going up and down vertically, it's the precursor MZ. So just imagine a very low MZ value, mass to charge value, and then it increases all the way up to very high MZ values. So very, you can kind of think about it as very small peptides at the bottom and then very large peptides at the top. And then along the horizontal axis, going left to right, is the retention time, that LC gradient that Sue had mentioned. So at the very beginning on the left-hand side, those are very hydrophilic peptides. And then on the right side, there's very hydrophobic peptides. So the first thing you might notice in this ion map is this gray. That's the intensity of the peptide at that particular coordinate in retention time and precursor MZ. You see there's a little bit of a trend, right? The density of the gray in the ion map kind of tends to increase in precursor MZ as you increase the retention time. Large peptides, which tend to have high MZ, tend to also be more hydrophobic than small peptides, which have low MZ. That's the trend that you're kind of seeing in this ion map. Now, if we were to do a parallel reaction monitoring experiment, we would pick predetermined MZ values. So that's on this y-axis, that's picking very specific MZ values that correspond to peptides that we know we want to measure. And then with and then in retention time, someone's walking their dog. It's very interesting. And then for retention time going across the x-axis, we might do scheduling. So Sue had mentioned scheduling as an approach, and this just means if we were unscheduled, then for that MZ value, we would monitor that MZ value. We program our mass spectrometer to look for that MZ value for the duration of our liquid chromatography gradient. Say we're running a 60-minute gradient, we would tell the mass spectrometer, monitor MZ value 1, 2, 3, 4, over and over and over again across the whole gradient. However, Maybe that doesn't make sense because we know our peptide should only elute at one spot. So we can tell the mass spectrometer, don't bother looking for this peptide at the end of the gradient because we know it's going to elute at the start of the gradient. That's all scheduling means. So it's just a way to squeeze a little bit of extra performance out of your instrument. So parallel reaction monitoring, you see in this ion map, we're not covering a lot of the space, a lot of the proteome space, because we don't care about most of the proteome space. We only care about very specific parts of the proteome. Sounds like a lot of people are also very familiar with data-dependent acquisition. So if we look at the same ion map and we look at a data-dependent acquisition, at every kind of chunk of retention time, we're only going to trigger precursor MZ at spots that are the most intense. So we tend to cover more of kind of that center ion cloud and don't tend to cover more of these outliers because we're only picking the top 10 most intense parts at any given retention time across this gradient. So finally, with a data independent acquisition, you see it kind of almost looks a little bit more like a parallel reaction monitoring, but tilted, just 90 degrees. So with a data independent acquisition, instead of scheduling the instrument for specific chunks of retention time, instead now we schedule the instrument for chunks of MZ, and then over retention time, just keep monitoring across that cycle of MZ. So it's more systematic, it looks more organized than a data-dependent acquisition. It looks similar in some ways to a parallel reaction monitoring, but covering more of the MZ space than a parallel reaction monitoring would. So with a data-independent acquisition, let's say if we were to take just one of these chunks, so this is a precursor MZ value, say this is 600 to 650 MZ. At this one retention time, we take an MS-MS scan. We program the mass spectrometer to make a mass spectrum for all the precursors from 600 to 650, say. Let's say this particular peptide happens to fall in that scan, we would get our MS2. But because we are monitoring that window, that MZ window across all of retention time, we can stack each one of those MS2s, each one of these rectangles is another MS2. If we stack all of those MS2s across retention time, and we looked down all of those, so looking kind of down through all of these blue boxes, we get the chromatogram. So now we can do everything chromatogram based for our quantification. This is super powerful compared to other DDA-based approaches because chromatogram-based quantification tends to be as sensitive or more sensitive than even those isobaric labeling techniques. Plus, now you can do more than 16 samples. You can do hundreds, thousands of samples. And it's cheaper because you don't have to buy the TMT kit. So that was kind of two ways for us to think about DIA. You could think about it at a high level as just a way to balance number of samples, number of proteins you measure and how sensitive they are. Or if you're more of a mass spectrometrist, you can kind of think about it as that ion map. Kind of depends which way you're approaching mass spec proteomics, more from a biological question side or from more of like an analytical chemistry side. There's a lot of constraints and compromises. So one thing that Sue had kind of mentioned is you're limited by your hardware. So you can only make MS2 scans as fast as your hardware can go. And for mass spectrometers, we measure this in hertz, so like a scans per second. You're kind of capped by your hardware. Related to this, in order to get enough points across the peak for a chromatogram to be good, precise, accurate quantification, you need at least 6 to 8 measurements. So you can kind of see how those two play into each other. If you need at least 6 to 8 measurements, but your instrument can only make so many measurements a second, you have to do some math to figure out what the best way to run your instrument is. There are some compromises with DIA. The wider you make those MZ windows, the more interference comes into your quantification. We'll talk more about that later and like actually just show you what that means. And then finally, you can't measure across the entire range of the proteome. You have to choose where you want to measure. So we're going to talk a little bit more about what these mean. So what do different implementations of DIA look like? I want to give you just a kind of overview of different flavors of DIA that you might see as you're coming across this type of research. So the first, kind of technically, parallel reaction monitoring is a type of DIA. So you have your precursor scan, let's say from 400 to 1400 MZ, and then you're going to program your mass spectrometer to collect just very specific fragment ion scans. Then you'll take another precursor scan, and then your mass spectrometer will collect those fragment ion scans again. So you're collecting the same fragment ion scans across multiple retention times so you can build that chromatogram. So you might get a mass spectrum that looks something like this. It's pretty clean. This mass spectrum looks pretty clean to me because there's not a whole lot of peaks. We'll be showing you examples of DIA spectra that are very complex. If you were to widen out your parallel reaction monitoring, instead of doing each one of these individual, very specific fragment ion scans, if you go to the exact opposite of this, it would be like an MSE, an MS everything. And your mass spectra would look something like this. So this is now, instead of taking just one very specific fragment ion scan, what would it look like if you took a full fragment ion scan for everything at that particular retention time? You can see it starts to get a lot more crazy, right? Instead of having just one very specific fragment ion scan, now you can see there's lots and lots of peaks in here. So this is one way you could think of two extremes of data independent acquisition, right? So you could think of parallel reaction monitoring as like the most specific you'll ever get, and then MSE as the widest, least specific you will ever get, either very, very narrow windows or very, very wide windows. So MSE, you might think, well, why don't we just do that? That seems easier than trying to figure out the precise Goldilocks fragment ion window width. It would be great because that's a great advantage, right? You get to quantify with your fragment ions using that chromatogram-based quantification. You would get very, very frequent scans because your instrument is just flipping through all of these fragment ion scans, and you don't have to compromise the range of MZ that you're measuring. See, the problem is you can't really analyze the data. You would have to try to come up with a computer algorithm to split that very complex spectra into its constituent peptide spectra. And that is very, very difficult computationally. So this approach is not typically seen too much now. There's a little bit of a resurgence in interest in building those computer algorithms to try to deconvolve these peptide signals or come up with a way to analyze this data. So it might be something you see in like a 5-10 years from now, breaking research kind of thing. But for now, anybody use this approach because it's just way too hard to try to analyze. So compromising between very specific PRM and very non-specific MSE, there are a couple choices. What about a PRM just slightly wider MZ windows than a PRM? This very narrow window DIA is the Pacific method. And you would tile very small fragment ion windows and continuously tile them across until you've covered your full MZ range. So this very narrow window approach gives you simple enough fragment ion scans that you can really basically use DDA analysis on it, but still get all the advantages of DIA for quantification. The disadvantage, though, is because you're using such narrow windows, almost no instrument is really fast enough to cover a whole proteome with this narrow window DIA. Maybe in the future, there'll be an instrument fast enough to do this, but for now, you would have to inject each one of your samples multiple times, multiple injection in order to cover the full MZ range of a proteome using these super narrow windows. So super easy to analyze, but kind of a pain because you would have to do these multiple injections for each of your samples. Okay, so the next thing, okay, well, what if we make them slightly wider, but not quite as wide as MSE and looking at everything? So a wide window DIA, swath, or DIA, they're the exact same thing. Swath is just a branded like trademark name for DIA. This is where pretty much everyone in the field is using their DIA these days. So it's a slightly wider scan, and you tile those across the full MZ range that you want to measure. Take your precursor scan and then tile them again. So now every cycle of your DIA, you're getting MS2s for every peptide across the MZ range that you're interested in looking at. There's some variations on how exactly to set this up, but this is generally what you'll see everyone in the DIA application method section doing, is some slight variation of this type of DIA. So there's a lot of benefits. You still get that quantification with fragment ions. You get your frequent scans, so you get enough points across the peak, but you're not quite as reliant on deconvolution as that MSE if you were to have super, super wide fragment ion scans. The disadvantage, you still have to pick what your precursor MZ range is going to be, and that is not necessarily the same for every type of sample, so that takes a little bit of method development to figure out exactly what range you need to do. And you still need to use special software to analyze this. You can't just use a DDA software for the most part. You have to use special DIA processing softwares. So advantages and disadvantages. So just to kind of reiterate, there's that super, super wide MSE version of DIA, and that spectra just looks super, super complex, right? If you compare that to these DIA swath methods, on the bottom, these MS2 spectra look a lot more typical for what we would expect to see a peptide mass spectra. Not quite so many peaks, and you can kind of, for those of you that are kind of in proteomics, you can maybe a little bit more by eye start to see the spacing of amino acids in the MZ range. So MSE would be super great. The computational proteomics isn't quite there yet for MSE, but maybe in the future. For now, DIA, definitely the flavor du jour for doing these DIA experiments. So kind of, I think this is the most popular DIA windowing scheme that I tend to see on Orbitraps. So if you're on a TOF instrument, I don't know quite as much up to date with TOF instruments, but you take each one of these fragment scans, you take 25 of these fragment scans, and each one of them is 24 MZ wide, covering 400 to 1,000 MZ. You take another precursor scan, and then you start all over. Another 25 windows of 24 MZ width. So now you've covered everything. If you think back to that ion map, the goal is to try to be covering the darkest part of that and intensity across the strip of MZ values. So we mentioned on your instrument, some of your instrument, you're kind of capped by the hardware, how fast you can go. For a Q Exactive HF, it takes about 2.5 seconds to go from one fragment ion scan at 400 MZ to cycle through all the 25 windows, take a precursor scan, and then get to the next 400 MZ. So in your liquid chromatography gradient, you would have an MS2 scan, you would have a point across the peak every 2.5 seconds. Okay, so you have all these fragment ion scans. Let's take, for example, just one of those fragment ion scans at some particular retention time. At a given retention time, that fragment ion scan gives you an MS2, a mass spectrum. But remember, we're taking multiple of these measurements over time. So we have multiple MS2 spectra as the peptide is eluting off the column in your liquid chromatography. So retention time is moving on. And as the peptide elutes, we take one MS2, and then the peptide elutes some more. We take another MS2, peptide elutes some more. We take another MS2. So we're getting these points across the peak. So now we build our chromatogram across all of these individual MS2 values. We will see chromatograms, see peptides start from low intensity, build in intensity, and then go down. But we do have caveats when we're doing these DIA style experiments. The first is that the ions This is more for like the mass spectrometry crowd. If you're coming more from biology background, this is a little in the weeds for you. You don't super have to understand the details of this, but for the mass spectrometrists, the ions aren't going to necessarily be centered, and your collision energy is not going to be optimized for each individual peptide. So for DIA, you have a fixed collision energy across your entire experiment. And the ions aren't going to be perfectly centered in this bucket of isolation values. So you might get some peptides that are kind of on the edge. There's almost always going to be multiple peptides in the window. And these edge effects, if you have a peptide that happens to be sitting kind of on the edge, the mass spectrometer, the quadrupoles, aren't perfect at transmitting ions. So towards the edge of the windows, you'll get decreased efficiency. So this is a good spot for me to stop and take some questions before we go to the next session. So Angel has a question. What is the resolution for this scan? The resolution you choose depends on your instrument. So we have a paper that I can put in the chat. I can have, I'll remind myself to put a paper in the chat that is, I'm the first author. It's MCP 2020, Acquiring and Analyzing DIA, blah, 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 blah. But it has a whole table for Orbitrap instruments that does the math for you and balances how wide the windows can be and what resolution to use to get that speed. Because if you increase the resolution, you're gonna take longer to get your points across the peak. Paul asked, do measurements for peptides that exist right at the MZ boundary suffer? Yep, exactly. That's that quadrupole transmission problem. We try to get around this, DIA researchers try to get around this by using what's called forbidden zones. So based on kind of average isotopic distributions of peptides, you can kind of theoretically calculate where there cannot possibly be a peptide ever. We try to place our edges of the windows where those forbidden zones are. So that's like a very deep in the weeds mass spectrometry answer. But yeah, those measurements don't end up suffering because we position those windows in the forbidden zones. Or you can. Not everybody does, but everybody should position their windows at those forbidden zones.
One thing we should point out is the forbidden zones are really for charge 2 and 3, 1, yes. 2, and 3. And if you go up to higher charge states, they work, they don't work as well. So
yep. But, yeah. And there's also caveats too. If you're doing something like phosphoproteomics, phosphorus makes a very different forbidden zone profile than the regular carbon, nitrogen, and oxygen. Very in the weeds answer, but I can throw some references in the chat or I'll throw references in that document link on the May Institute website so that you can have them after this Zoom gets shut down. So in the DIA scheme, does the software stitch together the fragment scans? It doesn't need to stitch together the fragment scans to ID the peptides. A lot of DIA software will look directly at the fragment ion scan and analyze it from there. So you don't need to use the chromatogram to ID the peptide. You're still doing the identification, the detection at the MS2 level. Are there benefits to doing slightly overlapping windows for DIA? Absolutely. If you use slightly overlapping windows, if you're on a TOF, having the margins on your windows is good. If you're on an Orbitrap, do not use margins. Instead, do a full stagger, a 50% stagger. So that is also discussed in that MCP 2020 paper that I'll make sure I link in the documents. Jonathan Wilson, 6 to 8 precursor scans are required per sample per peptide. Yeah, so not the precursor scans aren't as important as the MS2 scans. Precursor scans, there are some people that make their DIA methods with no precursor scans. And depending on the software you use, you don't need precursor scans at all. You can just go straight MS2 fragmentation scans, and you want 6 to 8 of those scans so that you get 6 to 8 points across the peak in your chromatogram. And the fragments, it's everything in that any peptide that falls in that 25 MZ wide window will get fragmented together. So you'll have a chimera spectra, you'll have multiple peptides in the same spectra. That's why it's kind of nice to use like a DIA software to be analyzing this because they're specifically designed to take that into account. So the point of overlapping window DIA, so depends what you mean. For margin overlap, the point is this transmission efficiency by having the slight margin overlaps. You don't have that transmission efficiency problem. The other way around that is to just use the forbidden zones and put your window boundaries at the forbidden zones. For the point of a staggered window DIA, you can use computer algorithms, computational algorithms that will deconvolve the stagger so you get twice as narrow windows. So we can talk more about that if people are more curious about that. That's getting a little bit more into the weeds of a very, very specific flavor of this popular DIA scheme. Do you keep shifting the windows and sequential injections to cover the whole proteome? That is for that passive method, the very, very narrow window. You would, each injection, do a different MZ value. For this most popular DIA windowing scheme, you do not do sequential injections. Each sample is just injected one time, and you tile across this for every sample. So sequential injections, yes, each method, each sequential injection would have a different method for a Pacific style narrow DIA window method, but for this most popular style, this wider window, you don't do sequential. Where are you fragmenting the precursors? Same place you fragment them for PRM. So if you're on a tripod instrument, if you're on an Orbitrap, if you're on a TOF, you just fragment them all together and take the scan of the full kind of bucket of all the peptides that gets fragmented. Yeah, as far as using MSE, the software, as far as like getting good quantum identification, using MSE as a trademarked word for DIA, that's totally fine. Using MSE as the method of having every fragment ion going into the mass spectrometer at the same time, I don't recommend. Using MSE as just Waters branded DIA, that's totally fine. Use a method like this most popular windowing scheme, this wide window. That's totally fine. Okay, I think we're just about caught up with Q&A. So we started getting a little bit into this. So since the spectra are a little bit more complex, how do we analyze DIA experiments? There's a lot of options out there, so I wanted to put up maybe a couple considerations that you might have. We're not really going to talk too much about libraries, besides that sometimes you need them. We're not going to get too much into the weeds there. If you want to talk more about it, we can talk more about it afterwards. Does the software let you look at your data? And is the software free? Maybe these are like the top questions I get when people ask me. So of those, there's a lot more options than just the ones I'm listing here. But these are, I think, maybe some of the ones that I see the most. So OpenSwath is a software, requires you have this library and it's free. It doesn't have a good way for you to interact with your data and visualize it. Same goes for Swath 2.0. That's kind of a commercial version. So it's not free. Skyline, which you're going to be doing tomorrow and Wednesday, does require you have some sort of library. We're not going to talk about different types of libraries, but it uses a library. You can interact with the data and it is free. Spectronaut, you can use a library or not, and you can interact with your data, but you have to pay license. DIA Umpire is another popular one, was maybe more popular 10 years ago than it is now, but it's also a free option for you. Encyclopedia, this is the one that I tend to use the most. It does require that you have some kind of library and is free, but you don't really have a good way of interacting with your data. And Diane, D-I-A-N-N, Diane is another very popular one right now that's free, but doesn't really give you a good option to visualize your data yet. So you'd have to pull that data into something else to visualize it. There's a lot of options. It doesn't matter so much what you pick, but one hill that I will die on is no matter what you use, you should look at your data. Don't just believe the matrix that the quantitative matrix that these software spit out. Don't just take them at face value. Go and look at your data. And this is why. So this is a screenshot from Skyline looking at a chromatogram. So retention time on the x-axis, the time the peptide is eluting off of the liquid chromatography column, and then the intensity that is measured by the mass spectrometer. Each one of the colors is a different transition for this one peptide. So transition is just the fragment. So the peptide, we break it in two parts, and that part is the fragment from the peptide. So each one of these different colors is a different peptide. This is an example of a pretty decent looking peptide. We're going to look at some more crazy ones with Brendan tomorrow and Wednesday. Just kind of orient you. So we looked at Skyline before, but this is just how this chromatogram is just one piece of Skyline. If you were to zoom out and look at the Skyline software itself, it looks like this. So on the left-hand side, you have all of the targets. You have to have these targets in the document before you do anything else. Skyline needs to know what it's going to look for, right? So this hierarchy goes protein at the top level, and then the peptide sequence, and then the MZ value for that peptide with a charge state. So the plus means it's a +2 charge state. And then all of the transitions underneath here with their MZ values. You see their +1s. So the chromatogram window, just a tip for you for later, if you happen to lose this, it's very easy to get the chromatogram window back. Here I'm showing just the MS2, so the fragment ions, because that's 99% of the time all that I care about. But you can also see precursor ions if you happen to care about those. There was a question from before about analyzing DDA data in Skyline. There is a Skyline tutorial for doing MS1 full scan filtering. Maybe I wouldn't like super highly recommend MS1 full scan filtering for your quantitative proteomics needs, but if you happen to have data that's DDA and you want to pull it into Skyline, you can look at it that way. And what we're going to be doing today, hands-on, is going a little bit more into what does it mean for a chromatogram to look good? Because as you're going through this May Institute, if you're sticking around and doing more of the statistics, garbage in, garbage out, right? There's no statistics in the world that will save a bad data set if the data set itself is bad. Can't massage biology into a bad data set. So some of the things that we're looking for in a good quantitative data set, the peptide signal should all co-elute. So you see this blue signal is not perfectly stacked with the red, orange, and purple signal. You want them all to be a perfect rainbow, all perfectly co-eluting. The peak shape, ideally, will be very symmetrical. This peak shape is not bad, to be honest, but it's not perfectly symmetrical. It has a little bit of a tail. Signal intensity, as you get into more complex data, you will start to see other places in the chromatogram that could be the peptide. These top three, you don't need to know anything else about your experiment besides having raw data from your mass spectrometer. If you're really looking to go deep, especially with DIA, you should have a library spectrum. Where that library comes from, there's a lot of options. We're not going to have time to go too deep into those, but feel free to ask more if you're curious. The correlation to your library spectrum is very, very helpful in knowing that you picked the right peptide. Similarly, the correlation to the retention time, whether that retention time is predicted or measured from a real library that you collected on your instrument. can be helpful in knowing that you picked the right peak. And then finally, if you are like super gold star, A+ mass spectrometry proteomics experiment, you'll have a paired internal standard that has a heavy lysine or heavy arginine, and those two should perfectly co-elute. So that if you look for your heavy standard and your light standard should be right there, perfectly co-eluting with it. That one, not so common with DIA, but very common with DIA, maybe even required for a lot of DIA softwares, is the correlation to a library spectrum and correlation to a retention time. So there was discussion about how good the data looked before. We're definitely going to be looking at much messier data now. But this is what I mean by interferences and why maybe the commenter was saying like, wow, this data looks perfect, but what about real data? This is more maybe what you'll see in real data. So where exactly is the peptide? So using these criteria, and we're looking at this, and this is what we're going to be doing hands-on, looking at this data, where exactly is the right peptide. Especially on the left, you see there's in the orange Y4 signal, there's a huge interference. And it almost makes it hard to tell that right here, a little after 13.5 minutes, there's a beautiful stack of all the other fragment ions in a very symmetrical, pretty looking chromatogram. But there's this giant orange signal interference in that. Similarly, another type of interference that you might see in your data, here's another example, just about at 18.5 minutes with an orange, a blue, a purple, and a red fragment ion all stacked and looking pretty symmetrical, except for this giant interference in the Y6 signal. So this is kind of what we're talking about when we're talking about messy data, and in DIA, you're more likely to see these interferences than if you were to do an SRM, a targeted SRM, MRM, PRM style experiment. Skyline is super good at detecting clean peak groups. So this is an example of a data set where it's very, very obvious where the peptide is, and Skyline picked it correctly. As data gets messier, it becomes more difficult to pick the right peak. And this is why, and it's not even like a Skyline specific issue, this is every software out there. And this is why I have to emphasize so strongly, look at your data. Don't just trust the quantitative matrix that gets spit out from Spectronaut, spits out from Diane, whatever you're using for DIA. Look at that data. I use Skyline to always look at that, but look at that data and you might find it has incorrectly identified something. And whether you're doing your own mass spectrometry for your own research or whether you're like core facility working for a collaborator, nobody wants to waste years chasing down something that ended up being just a misprocessed signal. So double check before you publish, before you give your collaborator any answer, double check that it's real and that the signal is convincing. Skyline has a lot of tools for you that we're going to use. And I only have a couple more slides before we'll do the hands-on. So I'll definitely get to the questions too. But Skyline has some tools that we're going to use that make it very helpful to quickly curate data, even across pretty large data sets. So here is a retention time screenshot from Skyline, where across the x-axis going left to right, these are all the different sample replicates for this experiment. So these are each individual sample run on mass spectrometer and the data pulled into Skyline. And then on the y-axis, we're looking at retention time. In the box is the start of the chromatogram, the start of that peak, and then the end of that peak. And if you look across, there's a little bit of wiggle room for all of these samples. Retention time isn't perfectly reproducible across different samples because each sample has a very slightly different matrix, and even slight differences in matrix will change the retention time just a little bit. That said, there is one sample that is very clearly an outlier. So this would be an example, as I'm looking at a data set, I would say, aha, this looks like it might be a mispicked peak. So I could click on that, look at that chromatogram and ask myself, do I believe that the software picked the right peak here? Or do I need to manually curate this and I, as the expert, know that the peak is actually over here by 31 minutes and not at 33.5? There's other tools in Skyline that will help you look at this. So this is a similar style of data curation tool where we have the replicates going across the left to right x-axis, and then the peak area percentage. So these are all of those different fragment ions. They should all, across all of the samples, they should go in roughly the same order. So if red is always the tallest, and then teal, and then green, it should always go in that order. So when you see across these samples, red is almost always the largest percentage, you see that there's an outlier right there, where that chromatogram clearly didn't follow that same fragment ion order. The rainbow was a mix of colors, and it should not have, the colors in the rainbow should not have gotten mixed up. So between the retention time and between this peak area percentage, this is a signal to me as the expert to go back and manually curate this data before I try to push it downstream for statistical analysis. So that was pretty much everything I wanted to cover before we do the hands-on. Active areas of research, in case people have questions about this, the best practices for doing PTMs by DIA. This is still like a little bit of like maybe not a super figured out field. Predicting retention time and fragmentation for in silico spectral libraries. We're not really talking too much about that, but super popular way to do DIA is with these in silico spectral libraries instead of doing the DDA yourself. As data sets get bigger, your computer might not be able to process them. You might run into this tomorrow or Wednesday if you're operating on a laptop, for example. So people are working on cloud-based resources so that we can process larger data sets. Then finally, if you're coming from the DDA world, you might be familiar with FDR and decoys. This is a pretty big active area of research for DIA because the way we make decoys for DDA doesn't work great for how we do DIA. So doing proper decoy generation and proper false discovery rate is something to always think about with DIA and a little bit of bleeding edge in the field. So again, this was a lot of information. Really the only 4 things that I hope you can do now. Key differences between DIA and some of the other acquisition methods. That there's different flavors of doing DIA. It's not quite as cookie cutter as DDA or PRM. The caveats of DIA and the data analysis, main takeaway there, look at your data. And then finally, we're gonna look at some actual real life data. So while I get spun up to do that, we will go through questions. It's a question about Skyline for dose-dependent analysis. I don't think there's any statistical analyses in Skyline for dose dependency, like a dose response curve or anything. So nothing inherently in Skyline for that. But if you're sticking around for the rest of MS Stats, that would be a good place to ask them. How do you do manual curation in Skyline? That is literally the hands-on that we are going to spend the rest of the afternoon doing. Perfect. And then a couple much longer questions in here that I will try to get to. It looks like Brendan is getting one of the transition ones. So I'll get to Mark's questions, questions and comments on fragmentation and DIA. If you are fragmenting in the ion trap that uses resonance ionization, which is automatically optimized. We do not do automatic optimization for DIA. Nope, this is all NCE, and you have a fixed NCE across all of your scans. This is also true in QTOF too. So you do not do any sort of collision energy optimization for DIA. You pick one collision energy and you fix it across all of your fragment ion scans. So the issue comes, and this is getting a little bit into the weeds, the issue with this fragmentation energy, this collision energy, comes with the fact that if you have a DDA spectral library and you did not acquire that DDA spectral library with a fixed collision energy, then the comparison between your DDA spectral library and your DIA, acquired fragmentation, will not look the same. The more those don't look the same, the more dissimilar those look, the worse the dot product will be when you compare them. And the worse the software that's trying to analyze your data, the worse score it will give that peptide. So if you are going to collect a DDA spectral library, collect it with one fixed normalized collision energy that matches the fixed collision energy that you're collecting your DIA with. So it's kind of that question of getting a library that accurately reflects the real samples that you're running, with not only the exact samples that you're running, but also the exact instrumentation you're running as well. This is not necessarily just true for DIA. If you're doing PRM and you happen to have different collision energies, or you're using a different instrument to acquire your DDA, then you're going to go on like your triple quad or something. That mismatch between the library spectra and the acquired spectra on your instrument can give you a low dot product when it was just a collision energy issue. So that was like very in the weeds, but we can go back into that. Question about what's challenging doing DIA for measuring PTMs. Part of it is the issue of isomers. Typically, there are co-eluting isomers at the same retention time, and how you deconvolve that signal can be very difficult. Another issue is libraries. If you're using in silico predicted libraries, there's not great ways of doing that right now. Maybe phospho is like fairly well figured out, but for other types of PTMs, it's not quite as well figured out. It also becomes difficult if you're looking at small PTMs, like an acetyl or a methyl. Those can fall in the same window as their unmodified form. And now are you detecting and quantifying the modified or the unmodified form? There's big, huge area of active research there. But at the end of the day, it's definitely, maybe I would be a little cautious doing DIA for measuring PTMs unless I had a paired internal standard so I could be absolutely certain that the quantification was correct. We have a couple more questions. See if I could do a quick one. Do you have to generate your own DIA libraries? A lot of people are using predicted libraries these days. I don't know very many people who are now doing paired libraries unless they're doing the approach that I tend to use, which is the chromatogram library based approach, which does generate a DIA library. Most people are using these predicted spectral libraries these days. They're pretty cool. Can DIA incorporate ion mobility data? This one may be a better question for Brendan, but Skyline can incorporate ion mobility data. I don't know if that is incorporated in the Skyline DIA analysis in the way that you happen to be thinking, but ask that again tomorrow or Wednesday.
Yeah, I'm here.
There you go.
Brendan. Yeah, definitely. I mean, there's actually a whole tutorial on, if you're thinking timsTOF, there's a whole tutorial on DIA process. You know, we have definitely worked with FAME's data and believe that, you know, if somebody, I'm not sure anybody's given us a FAME's DIA file yet, but I think that Skyline should be capable of doing that. Yeah, so we've been working with ion mobility data for a pretty long time, and Skyline has lots of functions and features. And so, yeah, if you get started with some aspect of DIA with ion mobility and you don't feel like it works well in Skyline, just contact us. We put a lot of
nice. Yeah, I know there's some really cool like scanning swath, like sonar style, where it like slides the window across. I don't know any software that can handle that particularly well, but that's a pretty cool approach.
Our understanding is that SciAx on their side is doing the work to make it look like normal swath to us. So we just process it like normal swath. But yes, under the they're sliding the windows and doing something more complex, demultiplexing, and then, but we don't ever see that data.
Yeah, I personally have never interacted with that data. It's cool. I don't have access to it. Okay, so we're going to get into some of the hands-on. So what we're going to do is open up making sure I know where all of my materials are. Okay, so you are welcome to follow along with me on your own computer, or feel free to just watch me for now. And these are going to be recorded, so you could watch me this time and then do it on your own. Or if you feel good and confident, grab the PDF and just go to town. And you can just listen to me in the background and let us know if you get lost. So in the linked tutorial materials, there's this group studies folder. If you unzip that and you dig deep down, you will find this rat_plasma.sky.zip folder. You could unzip this here and open up the Skyline file, or as you boot up Skyline on this start page, there's an open file. If you navigate to where that .sky.zip is, Skyline will unzip it for you. So you don't necessarily have to unzip the .sky.zip. You can just open up directly in Skyline. So I'm opening up that, getting everything opened up for me. You should have a screen that looks something like this. Maybe the most important thing you will learn this whole week is how to change the color scheme in Skyline. My colors of my fragment ions might be different than yours. To change that, on the top left, it's tools, options, display, and you have all these color schemes. So I'll switch mine back to classic for now, so that we're all on the same page. But this is probably the most important thing, so that when you make your posters with Skyline screenshots, you can have your pretty color coordinated poster scheme. A little bit of orientation. On the left-hand side, we have all of those targets. So remember, protein level at the top. If you hover the mouse over the protein, it'll give you kind of all the FASTA info. Then the next level down, we have the peptide. And then if you click on the plus sign next to the peptide, that will show you the precursor MZ value for that charge state. And then the fragment ions that were chosen and refined for this project. So we're walking into this project. The peptides, the target list has already been chosen. They've already acquired all of their data. And now we are kind of the data analyst, and we need to go through and analyze all of the data. So another kind of piece of orientation down on the bottom right, below the replicate screen, there's a 1 out of 48 proteins, 1 out of 125 peptides, precursors, transitions. This is kind of a nice way to get a quick overview of what's in your Skyline document. So some more orientation. You may notice over in the target window, for each peptide at the peptide sequence, there is a little P with what kind of almost looks like an antibody underneath it. Some peptides only have the P with the little like red antibody. Some of them also have a couple of sticks in their icon. If you click on one of those peptides that has the little sticks, you can click on any of them. For me, I'm going to click on peptide LGGEE, blah, 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 blah. That little icon means that there is a library spectra for it. So with Sue, we just looked at some library spectra. There's a couple different ways that you can view this. I'll show you the way I typically use, which is at the top, at the menu, select View, Spectral Libraries. So now you can look at all of the spectra in your document. And Sue was kind of showing us how typically you would want spectra that kind of span across the MZ range. That means there's like lots of fragmentation. You want good fragmentation for your peptides across everything. So you can look at all of your fragment ion there. You can also go to the top right, go View, Library Match, and that will open up a window for the peptide that you're on. And now as you click or as you use your arrow keys to select different peptides, it will show you the matching spectra for the peptides that have spectra. This first one does not have a spectra for whatever reason. Either this peptide actually isn't seen with DDA, or maybe the sample that the DDA spectral library was collected on, maybe this peptide wasn't present in that sample. For whatever reason, there's no spectral information for that first one, but the rest of them, you can kind of see each one of them. So these samples, already some of these came preloaded. Oh, I will double check the Q&A. Sorry, I had navigated away. A couple questions. How are peptide spectral matches actually matched? So the spectral library has already been searched with another software. You could either use within Skyline, MS Amanda is now available to search directly in Skyline and build a spectral library from that DDA, or you could use whatever your software of choice, Proteome Discoverer, Comet, Mascot, Sequest, whatever you happen to use, MaxQuant, and then you can build spectral libraries in Skyline with settings, peptide settings, library. You have these spectral library options. We'll try to drop some links to step-by-step tutorials in the chat. So now that peptide sequence is already associated with that spectral match. So this peptide sequence in Skyline has that knowledge, that prior knowledge of what the spectra should look like. So it doesn't have anything to do with the MS2 peaks. It doesn't have anything to do with points across the peak. It's just DDA. So was your DDA search able to find that peptide in your spectra? For single cell proteomic data, I don't have any experience with single cell proteomics or doing DIA with that. The spectral library, all I can really advise is the spectral library should be as representative of your real samples as possible, because the way fragmentation and matrix effects kind of act together with retention time, you should try to have it be, if your real samples are like nanogram levels of peptide, I would make my spectral library with nanogram levels of peptide. And now in Skyline, if you use inserted protein peptide instead of library, can you still view the spectra? Only if you already have the library set up. You could insert a peptide and it would associate that. But if you don't have the library set up, Skyline will not like make up a spectra for you. Okay, so it looks like we're mostly caught up. Hopefully everyone's just kind of like tabbing through the data, getting familiar with it. So we are going to just practice pulling in some data. So if you go to File, Import, Results, File, Import, Results, you'll get this Import Results window. So this is, I'm done acquiring a raw file or a WIF file on my mass spectrometer. You do not need to do any kind of conversion. Skyline will natively read across all the different vendor formats. You can even pull in different vendor formats into the same Skyline document. That was kind of like one of the reasons Skyline first was invented and like came to be. Was there was a big giant study and people were running different types of instruments, different vendor instruments, and there was no good way to analyze all the data together. So in this import results window, we're going to keep the radio button at add single injection replicates and files and hit OK. Now it's going to pull up a file browser window. Navigate to wherever your tutorial files are and go to the raw folder and click on just the first raw file. That's this D_102_rep1. Then you can hold down the shift key and grab the last one. So there's other ways you can do this. You can click and drag in the window. If you were a glutton for punishment, you can click each one with Control. I typically do the shift and then hit open. And now Skyline shows you what some instructors like to call Skyline TV, which is showing you all the chromatograms as they import. So Skyline, the way it's working kind of under the hood, all of these MZ values that you told Skyline to look for, it's going to look across all of your data for the scans that match those MZ values across the retention time that you set up in your settings. So if you're doing DIA, probably you would not choose to import everything across the full retention time, but maybe you are. This data is not DIA or DDA data. This data is SRM. So this is like a targeted experiment. Tomorrow you will be working with DIA data, and I think it has a DDA paired spectral library. I think this data is not PRM. I think it's SRM. I can check. Yep, SRM, selected reaction monitoring data. So there's more questions in the chat while my data is finishing loading in. How do you get a peptide match when there is no fragment spectrum in the library? So Skyline is actually doing its own kind of searching and scoring under the hood. It's taking all those MZ values and it's creating very similar score criteria from what DDA search engines do to find where the peptide is in your mass spec data. So Skyline does not require that you have a library in order to analyze data, especially this kind of data, this kind of SRM style data. It's nice to have because Skyline can use that as an additional scoring measure, and you can use it as an additional kind of sanity check. So as you're selecting peaks, if you don't have an internal standard, you don't have a heavy peptide, and the peak is consistent across 3 injections, is that correct? That would indicate to me that probably it is correct. You could also look for other potential peaks in your full chromatogram. Other peaks might look similar across your chromatogram. We're going to hopefully show you how to do that in just a minute.
So one thing, so there's been a lot of like, what if you don't have a library, but you know, and the data that Lindsay is showing, this data set at the time, we used just the public libraries and found that only about 50% of the peptides we were interested in targeting had library spectra. And so we started this experiment just targeting all tryptic peptides, and then these are the ones we thought we could detect. But we now, since then, we have PROSIT integrated, and you can actually, one of the things I now frequently demo here is you can see which peptides have spectra, and there are these little scratch marks on their icons, these little, you know, like look like a little spectrum right down at the lower right corner of the icon. Yeah, Lindsay's hovering the cursor over it. So, but you could get all of them to have something by going and building a Prosit library. And I frequently do that when working with this data now. It's just build a Prosit library, and then you have something where you can look at a .p-value, and we've proven that that's a really great option if you don't have a spectral library.
Yeah, totally agree. I do that a lot too when I'm building a PRM. Instead of making a DDA library or something, I'll just throw it into Prosit. So now we have kind of all of our data. I wanted to show you, there was a question last time about how to arrange the windows. So I'm going to close my library match. I'm going to close my retention time. I'm going to close my peak area so that I just have the chromatograms. And I just want to show you kind of how you can manipulate what Skyline looks like, because that was a question from last time. So first, you can click and drag and lock, like kind of dock any of these windows wherever you want. So for example, now I'm going to click on targets. I'm click and hold on targets. And you see now I have this like blue window. I'm click and holding, and I can drag that wherever I want. You see, as I'm dragging it, some of these kind of windows are popping up and give me the option to dock at this location. So over here on the far right, if I finally release that click and drag, now my target window is docked on the right-hand side. I'm going to drag my target window back to the left-hand side because that feels very weird to have the targets on the right-hand side. I never see anybody do that, but maybe that somebody is you. So now we are going to pull up the retention times window. So view, retention time, replicate comparison. And now we get that retention time window back. So let's click and drag retention time to the bottom. And then let's view peak areas, replicate comparison, view peak areas, replicate comparison. And that one already came up for me, docked to the bottom right where I was going to put it anyway. But you can click and drag and get that docked right next to retention times on the bottom. Okay. Now your window should look maybe something like this. Now for our chromatograms, we're going to do view, arrange graphs, grouped. So view, arrange graphs, grouped. We're going to group these in 3. We are going to select the radio button, distribute graphs among groups. Under sort order, we'll have document. And all of this is in the PDF. I'm on the PDF page 8 if you were following along. So it should look something like this. We hit OK. For me, I had it kind of already arranged that way, so it didn't really change too much. Looks like maybe we're still doing good. So you should have something that looks generally like this. Now let's start actually looking at some peptides. So the first peptide I want to look at is GILAA. So you can find, just like in a Word document, you can Control F and enter in a peptide that you want to find. You don't have to have the full thing typed in. Oh. And my document is not finding this peptide for some reason. That's okay. I can find another peptide. So when we're looking through a document and we want to analyze it, I had told you before that we were kind of looking for these peptides that might not follow the trend in retention time or the trend in peak area. So you can tab up and down through different peptides using your arrow keys up and down. That's typically what I do. Or you could use your mouse to click on them. So we find some peptide. I'm on peptide AFMD. To find that peptide, AFMD. Find next, and it should take you to that peptide. You could do edit, find, and type in that peptide, find next to find that peptide. You see this particular peptide, it definitely looks like maybe there's a lot of dirty signal in here, right? Like these look pretty awful. If I look across different measurements of this peptide, I can click on their different retention time boxes and it'll take me right up in the graph to where that peptide is. And this looks super messy. This is an example of a peptide that I would not feel very confident in. Where is this peptide? So if I click on some of these, one thing you can do is take your mouse, hover where it says retention time in a graph. You could either use a scroll wheel to zoom out and see the full measurement for this peptide. See how messy this data is? So the first data set we were just looking at earlier, that plasma data set was super, super clean, right? It was so obvious where the chromatogram was. And now we're getting into data that is a lot less clean to look at. So over the next kind of 5-10 minutes, I'm just going to show you how I kind of tab around and might check some of these things. So I might kind of peak area. I might normalize. If I right click in peak area and I normalize to total, now I get that peak area percentage that I was telling you about before. So remember I was saying retention time might not be perfectly reproducible across different samples because of matrix effects. The peak area percentage can give us another kind of clue whether or not we've picked the right peak across all of the samples. So see, even though retention time for this peptide is bouncing around between 18 minutes to 19 minutes, the peak area percentage is very reproducible across this. So we can look across a bunch of these peptides. And some of these chromatograms, this example on ENLSPP, this chromatogram looks great. If there was a chromatogram that did not look great, so we can go back to some of these guys down here that looked awful. So this peptide AFMD just looks awful. The retention time is all over the place and the peak area percentage is all over the place. So trying to decide which of these peptides is correct. It doesn't have a library spectra. How do you know which one is which? You might not, just period, know which one is which. But if I want to assume that this peptide should be around 18 minutes, what I want to do is integrate the area at 18 minutes. So these examples of outliers up at 21-ish minutes, those I would click. I would click that peptide. I would find where the window is, and I would zoom to the 18 minutes, and then underneath the x-axis, I would click and drag where that 18-minute is. This is so that I'm integrating the area. The quantitation is going to be with all of this same mess as all of my other samples. So I would try to align all of the retention times to about that 18-minute mark. Some of these are maybe too early. Some of these are maybe too late. But I would try to integrate the same area for everything for this super messy peptide. Some of these are very obvious and they look great and you don't have to do any manual curation. So if we just keep tabbing down and just look across these, a lot of them look awesome, right? And before we do our statistical analysis, we would just want to find any of these that look maybe a little messy. And see if we can make sure that their integration matches. That's kind of the data processing that I might do. Oh, there was a question, how do you plot the area percentage on the y-axis? Right click in the peak area windows, normalize to total, and it'll give you the peak area percentage. The dot product does not include retention time. The dot product, and I can drop a link in the document, a link from the Skyline support board that describes exactly the various different dot products that Skyline will calculate for you. It's just a factor of the library spectrum match. If you want to look at retention time variability, if you right click in the retention times graph, and then go up to the top opt-in graph, you can look at regression, score to run. And this gives you the SSR calcs. This is the predicted retention time across all of the different peptides in your document. You can also click on these. All of these graphs are also interactive across Skyline. And you'll see some examples here of you click on some of these points and it'll bring you right to where the peptide is or where the peptide should be. So this is another example of curating your data. So here, this particular peptide, you can see in this middle graph that I have, so I'm on peptide IFPENNIK. This peptide got truncated. So this would be another example where I would manually want to curate this peptide and take everything from the truncated form of that peptide. Maybe this is a peptide because it's truncated. All of my statistical analyses downstream are going to be messed up because I don't get the full peak. So maybe this is an example where I wouldn't want to push this into a downstream statistical analysis because it's clear that the measurement has an error. This peptide has a measurement error. If I try to push this downstream for statistical analyses, then I'm going to kind of waste some of my multiple hypothesis correction that you'll be learning about later. But I'm going to test this peptide and I know there's going to be this high variability because it's the measurement itself has a problem. So this is why with DIA, with anything, I would flip through every single peptide that I would want to follow up on and convince myself that it's real. So at the scale of DIA, maybe that's not practical. In this document, we only have 125 peptides. So that's reasonable to spend, you know, an afternoon with a beer, going through your Skyline project and curating your data, looking for all of these issues with the data. For example, this mispicked peak right here on peptide DYVSQ. This peptide, it's clear in this file that the right peak was not selected. It's another one of these truncated.
Lindsay, we have a hand up.
Yes.
Lee Foster.
Hi, can
Oh, sorry. I didn't realize there was another host that could unmute. I unmuted you again. You should be good.
yeah. Since you promoted me, I have the power to unmute myself.
Yes.
So I missed the part where you changed your retention times graph to score to run regression. Could you just show me how you did that?
Yep. So right click in the retention time graph.
Mm-hmm.
And then in the pop-up, the top opt-in graph. And then regression, score to run.
Okay. Okay, that's very cool. And I didn't know that you could do that. The other question that I had is when you have your peak area percentage and you've got it normalized, you know, it's very easy to tell when you pick the wrong peak. But I see you doing some kind of wizardry where you're able to eliminate those peaks that are not correct, and you're able to get all of the samples to align at the correct retention time. So how are you doing it where the ones that are wrong, you're just aligning all of them to the correct retention time, so it's so easy to find that peak?
Yeah, so maybe there's a couple things that I have enabled. So in the chromatogram window, if you right click in the chromatogram window, any one of them, it doesn't matter, you should get a pop-up that down towards the middle, there's an option, synchronize zooming. If you click that and select that, now all of your windows will zoom at the same kind of rate. They'll all be scaled the same.
Okay,
that is a piece of wizardry. This is why I try to do a little bit more unstructured Skyline at the end.
Just be careful on that one. So Lindsay, right click in there again in the chromatogram. I think you have the y-axis auto scale turned on for best peak. So the y-axis isn't changing when you do that synchronized zooming. You have to turn off the auto scale for the X and Y in order for it to be like everything adjusting properly at the same time. Like everything, both dimensions.
Yeah, my y-axis scale is not the same between these. So yeah, that is, if you hover your mouse and zoom on the y-axis, you'll see the intensities now are scaled. If that ever happens and you want to reset it, I always just go to another peptide and then click back and it resets. But here's another kind of example where as I'm curating my data, nothing got picked in this one. So I would just click and drag. So this is not maybe feasible for a targeted experiment. This is totally feasible. And if you're not doing this, shame on you. You should do this. For a DIA level experiment where you have, tens of thousands, hundreds of thousands of peptides, not feasible. In that scenario, maybe you would do your data processing, you would do your analysis, and now you have your list of maybe a couple hundred significant peptides or proteins. Those I would put in Skyline, no matter what DIA data processing you're using, whether you're starting in Skyline for your DIA data processing, or if you're using Diane, Encyclopedia, Spectronaut, whatever you're using. Once you have your list of significant hits, put those in Skyline and convince yourself that they're real and that the software that you had used had actually picked and processed the right peaks. So hopefully this gave you kind of a little bit of a deeper look into using Skyline and set you up for the DIA day tomorrow and Wednesday. I think we are supposed to be done now. So I will stick around a little bit, but thanks for joining us this first day of the Northeastern University's May Institute for Computational Proteomics. We'll stick around if you have some questions. Check those documents on the course website for any updates or references. We'll try to make sure that all that information gets in there. But thanks for joining us today, and good luck with the rest of the week. We'll try to answer any last questions you have. Thanks, everyone.
So maybe I'll just ask one last question.
Yeah.
So I mentioned in the Q&A that most of the time what I'm using is like a DDA to PRM workflow. And one thing that I always struggle with, and it confuses me every time, is the importing of the library and how you keep it separate from the actual data set that you're using, and you're just trying to reference that library. So could you just kind of elaborate to that workflow? Because I'm still trying to get it straight in my head.
Yeah. So you're having trouble. Do you mean like you'll get your spectral library in and then it'll try to like add every single peptide that you detected in your spectral library to the document, and you don't want that?
No, I've got a pretty good workflow for designating which peptides I want to look at and which transitions I want to look at. But it's just confusing for me to try to put the library in there and then and then Skyline is trying to analyze my library. And I don't want to do that. I want to apply the library to get the right spectral matches on my PRM data set. And I get confused.
Yeah, so it should, if you do settings, peptide settings, and go to the library, and you already have your spectral library, you should be able to do edit list and add. And I'm trying to find where the tutorial is that I usually use for that.
Yeah, if you could just point me to a tutorial, that would be really helpful.
Yeah. I think it's in this one. I'll put it in the chat here, but I'm also going to put it in the document. So that everyone has it. Building spectral libraries. Skyline. Yeah, that's typically what I do. And it looks like we got everyone else in the Q&A. I will try to drop as many of these links as I can remember into the document for the course. And yeah.
So Lee, when you're importing or building a spectral library, if you import it, it should already have this .blib file extension on it, in which case Skyline isn't really doing much other than like opening it up. If you're generating or building a Skyline a spectral library, then you're going to be pointing to search results. That's Proteome Discover is going to be .pd results, or might be an MZXML or some kind of other result file, in which case then it will build it. And as it's bringing in the data, you will see it sort of what looks like analyzing as it's importing and opening it up. It's not really changing anything. All that it's doing is like building these spectra and labeling them for you and then matching it to what's in your peptide list. So there isn't really an analysis that Skyline's doing with it. It's more of just linking up what results are present and then what's in your Skyline file. If it's doing something different, I don't know what that would be.
Yeah, and I will say, if you do have, it happens to be like a specific issue with your Skyline document or your spectral library, the Skyline support board, I just dropped a link in the chat and I already had it in the document. The guys are super responsive. They'll get back to you very, very quickly. They can help you out if it's like something weird with how your Skyline is exactly set up.
Yeah, thank you very much. I had another question. You kind of piqued my interest when you said that you use mostly these synthetic libraries. All the stuff that I've done has been experimentally derived. So do you have any experience comparing those two sets of libraries? And what would you say are the caveats of doing it completely synthetically?
Yeah, so I will say I have been very impressed with the predicted spectral libraries. They, in general, perform just as well or better than DDA.
Yeah, I did a presentation. You can find it on the Skyline website. I can post it into the. I presented at ASMS a couple of years ago, comparing PROSIT to empirical spectrum, even for a QTOF, triple TOF, SciAx triple TOF, and also for a Q-Orbi. And yeah, the spectra worked really well. It was impressive.
Yeah, I do know when I was looking at like the synthetic spectra, this was quite a few years ago, and I know there's been a lot of progress in this particular area, and I've heard that it's very good. I just haven't tried it myself. So, I mean, I guess I'm just wondering what the limitations of it are. Like, one thing that I find is that you have your theoretical peptide and you use it like as an internal standard, and then you learn that it either doesn't ionize well, or that the linearity is poor, or the LLOQ is poor, and you will only derive that experimentally. Is that something that they've gotten better at, or is this something that?
No. Any of those analytical figures of merit, you're not going to get from a predicted spectra. The predicted spectra will literally just be a spectra. It won't tell you anything else about the peptide, like linearity or LLOQ or ULOQ or reproducibility. It won't necessarily tell you anything like that. It'll literally just give you a dot product for how well your acquired spectra matches the predicted spectra. So definitely not, definitely hasn't gotten as good at that.
They're adding
Okay.
models, but I haven't heard. They're adding models like the Prosit team has told me about some new models. We'll probably try to integrate this summer, but they tend to be for like charge state prediction and still like things like and whether or not a peptide is likely to even just show up on a mass spectrometer, but they haven't gotten to like predicting whether or not the peptide is going to be linear. And we may never. We'll see. Okay, so here is I just put in chat a link to the presentation where I talk about how Prosit for the spectrum model and the retention time model, how it does compared with empirical data.
And I added that link in the document for this session.
Okay. All right.
Yeah, thank you very much. Appreciate it.
Nice job, Sue and Lindsay. Thanks.
Yeah. Yep, and Ivan, I added that Prosit video to the document, so you should be good. NIST peptide libraries, you may have noticed in Skyline, if you go to settings, peptide settings, spectral library, there's a direct link straight to the NIST peptide libraries. So you can definitely use pre-configured, like downloaded spectral libraries from Peptide Atlas, from NIST, anywhere you can find a spectral library, you can get those into Skyline and use those. With the caveat that if it's not your exact instrument, with your exact instrument settings, you might have that issue with the dot product and the spectral library not matching your acquired spectra. Comments related to Prosit generated libraries do also apply to Spectronaut or Diane library free. And this is just another one of those things where, like, no matter what you're using, Spectronaut, Diane, Skyline, whatever, look at the data before you send some poor grad student off on a wild goose chase to characterize some protein that ended up not being real mass spec signal. Just check the data. I think we're about 10 minutes over now, so I think I'm going to shut down this session. But join us again tomorrow, same time, same place. Brendan will introduce you to getting more in depth with DIA and data analysis using Skyline. So hopefully it won't be quite so much of a crash course in Skyline after seeing it a little bit today. Thanks everyone for joining us.
Thanks, everyone.